Hello, everybody. Welcome to today's NCBI webinar. I'm going to be talking about some enhancements to NCBI BLAST and Primer BLAST. This is Peter Cooper. If you have any questions, you can write to me directly there at peter.cooper@nih.gov. And I'm one of the people at the NCBI service desk, and I'm also one of the people that supports BLAST. Wayne is as well, and Rana also works for the service desk. So we're just going to point out the Web BLAST services that you probably all know about. We're talking about some new features of Primer BLAST, where it can use multiple templates and you can link to the Primer BLAST service from the assembly database. We have some new description table options that we'll use today. And I want to talk a little bit about some condensed databases, one of which is available, the RefSeq Select database, and one of which is an experiment. And we just want to get some feedback from you all about that. And I'll have that in a survey at the end of the webinar. So you're probably familiar with this. This is the NCBI BLAST homepage. The top section is where you would go to do typical BLAST searches against databases, nucleotide BLAST, translating BLAST, protein BLAST. Then there are a bunch of specialized services, and the one we're going to talk about today in this talk is Primer BLAST. This is a primer designer and specificity checker. So it designs primers, and then it will check them against the BLAST database, essentially to see if they're going to be unique to your target sequence. Okay, so the first thing I'm going to talk about Primer BLAST first, and then we're going to talk about what's those changes that we've made to ordinary BLAST, and then talk about the databases. So we have the option to use multiple templates, and we also have these genome assembly specific databases. And basically, this is just a screenshot of the Primer BLAST interface here, and you can see basically what you do is provide a template sequence. And in this case, the new feature is you can provide multiple templates that are similar to each other, and then design primers for those against some background database. And this is actually one that we demonstrated a few weeks ago when we did a webinar about non-model organisms. What I'm doing in this case is I'm designing primers for a number of different species for a similar target. The other thing you could do is to take some splice variants, and that's the one we're going to do today, and design primers that would amplify all those splice variants. Basically, your primer results over there on the lower right show you you've got all these primers, and they were designed to avoid regions in that alignment you see at the bottom where there are a bunch of mismatches. So this is a pretty useful tool to make some primers that'll amplify a number of different sequences from different samples or from different organisms. The other thing you can do with this is you can set that specificity database to be a genome assembly. For example, if you had a set of primers that you designed that could amplify something from one species, you can check them in one of these partially assembled genomes like this one. This is for the humpback whale. So I could click the run Primer BLAST here. I can also blast the assembly from this position on the assembly database. But what this does is it sets that assembly as the specificity checking database. So it's going to take this pair of primers that I'm going to run. This is using Primer BLAST to check your primers against this humpback whale genome, and I can see that it does match, although not perfectly, to a region on that assembly. And that would be basically, that's going to be this gene, which happens to be the myoglobin gene, that I could then go in and try to get out of this partially assembled genome. We'll do an example like that in a few minutes. The other thing that you may have noticed is that there are new options on the descriptions table. That's the top portion, or really not the top portion anymore, but it's the table that shows you your matches on the BLAST output. So this is your typical descriptions table, and I've added some of the non-default options here now that are very helpful. So you can see that you have the ability to add scientific name and common name for the organism, the tax ID, and the accession link. Those are the things that are new. So check that out. It's very useful if you want to quickly scan to see what organisms you have in there, which is what we're going to do in an example later on. You might not recognize the scientific name, but you might recognize the English common name for that organism. You can also download this table in comma-separated variable format so that you just load that directly into Excel if you want to, which is pretty helpful. The other thing we're going to talk about today is some changes that relate to making what I'm going to call condensed or concentrated databases. Originally I wanted to call these reduced databases because they're reduced in size, but that doesn't quite capture what we want to talk about. Because really we're trying to get just some core information out of the database. So The problem that we're having, of course, is that the databases are growing at a tremendously fast rate. And so some of the new sequences that are added are not particularly adding a whole lot of information. So we're going to talk about two sort of databases that help with that problem. One is called RefSeq Select, and the other one is this clustered non-redundant database that I'll talk about in just a moment. Let's see how that works. So RefSeq Select is a project at NCBI. It really has two aspects to it. There are transcripts that are being produced for mouse and human only. This is part of a larger effort called the Match Annotation from NCBI and EBI, or called MAIN. But basically, what we're trying to establish is a single transcript and protein to represent each gene. So that's going to get rid of all the different splice variants that you might have for a particular gene. It's going to make a much cleaner set of results when you search BLAST with it. So that's why it's kind of helpful. The other one, which is kind of a different thing, but it's sort of part of the same project and the same database at NCBI on the protein side, is we're making a set of prokaryotic proteins from NCBI reference and representative genomes. This is about 13,000 genomes out of nearly 1 million prokaryotic genomes that we have at NCBI. And they represent important taxa, or every species that we have available. If you want to learn more about that effort, you can take a look at the links that are on this slide here. But they provide some reduced database sets. So here's what they would look like on the pull-down list. So if you look at RefSeq Select for the protein database, it's about 26% the size of, or the length really is what I'm trying to say here, because this is about the number of sequences pretty much, 26% of RefSeq protein, so it's much smaller. And for the RNA sequence databases, it's about 0.1% the size of RefSeq RNA. So these databases are going to be, you're going to be able to search these much faster than you could search the parent database. A related idea would be to cluster sequences at a certain percent identity, and we've started to do some of that to look at that as a way to make your results a little bit easier to interpret. So right now we have an experimental database that's clustered at 90% identity and overlaps at least 90% the length of a longer sequence. That makes a less redundant database. For example, one thing that would often happen here is it will collapse all the higher primate sequences into one sequence for searching. But that's useful if you want to expand your look-back time. To do a BLAST search that's takes too long, that doesn't take too long, and has a small set of results, and you can see, hey, does this protein get conserved all the way back to the base of the vertebrates, for example? Because right now, when you do that kind of a search, you're not going to see past the mammals for the most part with the number of sequences that we show you. This database at this clustering level is about 41% of NR, so it's going to be much faster to run. So what I'd like to do with all these things we talked about is to do some examples. So we're going to design some primers for the splice variants for P53, a human gene. Then we're going to check the primers against the Japanese macaque assembly, and I just put a picture of the Japanese macaque over there. And then we can take that macaque assembly, we can find the contig that my primers hit, and then I can use the RefSeq Select to basically annotate the P53 gene in that contig. And then I'll show you an example with RefSeq Select protein, just to show you the difference between searching the mutl protein from E. coli against NR, not against NR, against RefSeq protein versus what that looks like with RefSeq Select. There's another example in here that I don't think we're going to have time to do, but it's in the handout that's on the FTP site. And you can also get to it if you want to. And Rana, maybe you could put this in the chat, that compressed URL there. That should take you directly to the text file that has the accession numbers and other things you need to run these BLAST searches. So that second one, the animal ABC transporters, we probably won't have time to do that today, but you're welcome to try that one yourself. And I'll show you in a minute. They have preserved BLAST URL, so you can always load those. And then the last thing that I definitely want to show you today is what it looks like to do a clustered database search with human apolipoprotein E, so we can find homologs in other species. And we'll take advantage of that common name column that you can get in the descriptions table to take a look at our results. So what I'm going to do now is to switch over to a web browser. Okay, so this is the NCBI homepage, and the first thing we're going to do is let's go ahead and get P53. we'll take advantage of some of the little search shortcuts that we have nowadays on our website. So if I type in the symbol for, or a name, if you want to call it that, for a human gene, I do a search with it, I usually get this little device here that we've called at NCBI, we call this a known item search. Obviously, you're probably looking for this gene, and we gave it to you right there. And so you can see that this gene has 15 transcripts. These are different splice variants of the gene. Let me just open this panel here to show you something. So notice that out of these 15, we picked one, this one right here, to be that RefSeq select thing. And we'll come back to RefSeq select in a few minutes when we do a search using a config we're going to find. Right now what I'm going to do is I'm just going to get these sequences. So there's 15 of them here. Okay, and there they all are. These are all different splice variants of this gene. And let me just get, a convenient thing I can do here is to go ahead and get an accession list. And we're going to use that accession list in Primer Blast to design primers for this whole set of splice variants. I can just go ahead and select and copy that to my clipboard. And then I can just go over to the Primer Blast page. I'll just go to Blast. And then I can click here to get to Primer Blast. And then I'll just paste these in. Now, before I do that, I've got to remember that I'm not using one template, right? I'm using a group of sequences. So I'll just click that tab, paste these in here. So I have the 15 nucleotide accession numbers in there. And then on Primer Blast, so these are all the primer designing parameters. We won't really change any of those right now. And then I can look down here and I can see what my database is. So basically I'm comparing this to the human RefSeq RNA database. I want to make sure that these primers are going to be unique to those targets, the ones that I set up there. And then I can just press Get Primers to run this. Now this takes a little bit longer to run than I want to wait while I'm talking to you all, because we'd have a lot of sitting around time. So what I'm going to do is I've saved the job ID, and I can go ahead and just load that result. And you can do this too with your own jobs. They don't persist forever, but they persist for a day or so. Okay, and so you can see this is the same kind of result I showed you in the slides. So here are all the templates here. There are different splice variants. There's places where there are gaps. There's a longer one here. Not any mismatches, which is what you would see if they were different species. But I want to make sure my primers don't land in any of those gaps, and they did. They didn't. The other thing that I can see here that's interesting is some of these primers are going to be on different exons. Some of them are not. So primer pair 7 actually, I'm going to zoom in, actually is completely within a coding exon. That means it could work with the messenger RNA sequence, cDNA library, or it could work with genomic DNA. And so that's what we're going to use in a moment to try to find this gene in that macaque genome. So what I've done to make life convenient is I've taken these primers here and copied them so that I have them in that file. And like I said, that file is at the link where Rana posted in the chat if you want to look at it. So what I'm going to do is go back to Primer Blast. Not go back to Primer Blast. I want to go to the assembly database first, because what I want to do is to load the assembly that I want to use as a database in Primer Blast. So let me go back here to the NCBI homepage, and I'm going to go to assembly. And I'll just run a search here. So this is an organism search to find all the primate assemblies. And what I want to do is to find ones that don't have annotation, because we're going to do our own annotation here. Be able to spell and type. And this is a filter that you can apply to a search in that database. Okay, and we're going to pick, I'm going to use this genome, this assembly here, this is Macaca fuscata, which is a Japanese macaque. This is a old world monkey. Macaques are not that far removed from the lineage that gives rise to the great apes and humans. So these human primers that I designed, they probably will work in this assembly. So we can try that out. So I'll just load the assembly record. It's just a whole genome shotgun assembly. It contains a whole bunch of contigs. And I'll go ahead and click the Run Primer Blast button or link here. That will toss me over into Primer Blast. And now I can look down here and see that that's now the database that I'm going to be checking the primers against. So I'm going to put my primers in there. And like I said, I've got them over here in a convenient place. My sequences. There's a forward primer that was designed by PrimerPlast for the P53 human transcripts. And then I can go ahead and tell it to, it says get primers, but what it's basically going to do is to see what those primers match in this particular database. So this is using Primer Blast in a different way than we used it before. We're not designing primers, but we're checking primers against the database. And again, I don't want to wait that long for this to run. It takes about a minute or so. So I'm just going to go ahead and retrieve the results for us. Okay. Okay, and so there's a nice perfect match to this particular config right here. So, and there's also another config that doesn't match perfectly, but we're going to figure that this is probably the macaque P53 gene right here on this config right here. So I'll go ahead and retrieve the config. It's not too large. It's 27,000 bases long. And so if I want to, now I can sort of, let's try to use that RefSeq select database to annotate, to find the equivalent of the human P53 gene in this monkey genome. So I can just click this over here. I'm going to go ahead and just grab this sequence because sometimes when you go to BLAST, it has settings that you don't want, and when you change them, you might lose your query sequence. We'll just see what we get when we go over there. So I just click Run BLAST, it throws it in there, and actually it's not bad. That's what I wanted to do. So this is the macaque config. What I'm going to do down here is change this database now to RefSeq select, right? And I don't care what should be in there. Should be mouse and human sequences. I don't really care about the mouse one so much. So I can go ahead and just type human in this organism box here. We can grab Homo sapiens. That's going to limit me to those transcripts. And one thing, this is just kind of a useful thing to be aware of. This is a monkey genomic DNA, and monkeys and humans have interspersed repeats that are related to each other. And so it makes a big mess if you don't mask that kind of sequence when you're using a piece of genomic DNA. And this is just a useful thing to remember. If you're searching with a big piece of vertebrate genomic DNA, you want to do species-specific repeat filter. So I'm going to check that here. And this is the human filter, but that'll take care of the macaque repeats too. There is a macaque filter there too, but it's the easiest one to find right there. And then I'll run this. And again, I'm not going to wait around for this to take place. Sometimes it comes back before I manage to copy and paste my URL, we'll see. I did. Huh. Okay, that's fine. And sure enough, actually it turns out we got another gene that looks like it's probably correct, that matches this cDNA as well. And if you want to see this, just a good way to look at this alignment is just this graphic summary, because it gives you a really nice cartoonish view. Here's the P53 gene. These are the exons of the P53 gene. If you want to display that in a more interactive way, you can just click on graphics here. And it's going to load that config into the graphical sequence viewer. And you can see the matches here. And there is basically, it's aligned this human cDNA against this unannotated macaque config. And that's pretty much a good guess of what the p53 gene looks like in this organism. Okay, so let's do a couple of things with the RefSeq select protein databases. Really, we'll do one thing with that. And then I'll show you the cluster databases, and then we'll throw it open to some questions. So one real dramatic way to see what the clusters database do for you is to work with bacterial proteins, and so that's the one I'm going to show you. So let me take a bacteria. This is E. coli, which is one of the bacteria that we have a tremendous number of assemblies for and tremendous numbers of proteins for. When I do this kind of a search with that known item search thing, it actually gives me the one from one of the reference genomes. And so I can get this RefSeq protein here. And I could do the same thing we've been doing. I can run BLAST with it. And so what I want to do is show you what happens when I run it against a RefSeq select versus what happens when I run it against a RefSeq protein. And I won't bother to launch those. You already know how to do that, and we've shown you how to do that. So let me just show you what RefSeq protein looks like. And the thing about it that's, this is only 100 matches, so I could ask for more than that. But notice we never left E. coli, right? So everything in this result is E. coli. So if I'm looking for homologs in other species of bacteria, I can't do it. There's just too many E. coli proteins in here. When I do the same thing against RefSeq select protein, I can do a little bit better than that. So let me show you what that looks like. If you're trying to find homologs outside of E. coli, then that's the way to go. And so you can see I pick up other Enterobacteria here, Enterobacteriaceae like Citrobacter and Klebsiella and all these other things here. So this is a reduced database or a condensed database, but now I can at least get to see the different bacterial genera, which I couldn't see before. All right, so the last thing I want to do real quick with you is to just have you take a look at what happens with the clustered database. And I've already loaded those results. This is a page that is not, we don't want people using this just yet. Basically what I'm going to do is to take the human ApoE protein here. I took the RefSeq select version of it. And I'm going to search against NR. I'm also going to search against this clustered NR here. And I'm limiting this to jawed vertebrates. And what I want to show you is how much we can increase the look back time when we do that. So I've got the two results here. And these links are in your handout too. So here we have the results against NR. And what I've done is I've activated the common name so we can see the names of the organisms here. And you look and you notice that as we get down here, we have to go a long ways before we even get out of the primates. So the first non-primate sequence in this output is this one right here. That actually happens to be a bat. It doesn't have a common name. Here's a rodent. There's a woodchuck. Here's another marmot. And you pick up some other primate things. It depends on how complete the sequence is. But you really don't get very far. In fact, you don't really get past the mammals here. You go to the bottom of this, the only organisms in this list, there's one exception because there's a contaminant in here, but I won't bother you with that. These are all mammals. So we never saw anything beyond the mammals. That's not because there's not homologs, it's just that there's just too many mammalian sequences in there. And you might not be interested in all these primates. Maybe you want them to all collapse. And so that's what the clustered database does for you. So if I do it against the clustered NR, I start to see some different things going on here. So quickly, I'm getting down into other kinds of organisms here. So we can look for things in here. For example, if we wanted to find, let's say we want to find some whale sequences. Should have found some. Oh, you know what? That one got collapsed, so we won't do that. But let's, because whale is on the other one. Spelling was helpful. So you can see that we're outside of the mammals. Here's some fish. This is a goldfish. So I found that by the common name goldfish. If you want to look for the whale sequence, it's over here, which is what I meant to show you. So we can find the sperm whale over there. We don't find the sperm whale over here because, remember, these sequences are collapsed and we're only showing you one representative of it. And so there has to be a way for this to be useful for you to expand these and find all the sequences that are part of that collapsed cluster. And if you want to look to see the different organisms here, I just want to make a plug for the taxonomy report. It shows, it's a good way of seeing what organisms are in here. And I've gone all the way down to the taxonomy version. We can see we're at the base of the jawed vertebrates. So I can close up things here if I want to. I've already done some work on this, so you can see, I can expand this to see the tetrapods, including all the mammals. And then here we have some fishes. Even we have some cartilaginous fishes here. So we got all the way to the base of the jawed vertebrates by using that clustered database, and only 250 descriptions. Okay. So that's my demos. Let me just go back over here to the slides. And this last slide has some URLs for things that you might find helpful. And notice we have an NCBI Insights blog. That URL is on here too. So go there if you want to learn more about a lot of these features I showed you today, because we've got little articles about them there. Blast News is another way to learn about releases that have features like this in them. So let's open it up, Rana, to questions.
Great, we have a number of questions. Let's go ahead and start off with templates. One of the viewers today was interested in the primer blast where you showed that you could run it against several sequences. And he was interested in, is it possible to run primer blast against several genomic assemblies all at one time?
Yes, it's possible. You can't do it in the way that I did it today. So there is a way to, let me go back to my web browser here. Let me get a tab open that has a primer blast in it. So way you may need to help me out here. There's a way to set a custom database here. I can't remember exactly how you have to activate that.
Yeah.
Yeah, there
you go.
So you could potentially put some accession numbers down here to use more than one assembly. It will not take assembly accessions, unfortunately. So if you had chromosomes from a particular assembly, you could put those in here. Just a warning that it could quickly get too large for this to work, because the blast portion of a primer blast search is sort of a wide open search, and so it's very expensive to run. It consumes a lot of resources, and so it may just get out of hand real quickly if you do that. But it's possible to enter accession numbers down here. And don't forget you need to change your organism down here to what you want to use. So it's possible, but it's probably not as deluxe as you would like it to be. And part of the problem is that the assembly databases are going to get quite large if you start adding a lot of them in there.
Thanks, Peter. We have a series of questions about primer blast specifically and primers itself. So I'm going to kind of go those in order, and then we have a couple of just application questions. One of them, let's start off with designing primers in general in primer blast. And here's just a general question. What parameters should one keep in mind in order to design a good primer pair? Are there things that they should consider when they're looking at the primer blast page?
Well, I think the defaults are probably your best option. And that's why they're set that way. You can mess around with so what you're looking at essentially is sort of a hybrid application. So this top part here, these are the parameters that a program called Primer3, which is a public domain program, or somehow we can use it. We've incorporated it here. That's what designs the primers. And so you can change your TMs and all that stuff, and that's all the kind of stuff that you do for your PCR. These are optimal ones here. You can change your product size. Again, these are sort of the product sizes that most people work with. You know, the bigger your product, the less yield you're going to get in your PCR. So as a first pass, I wouldn't change any of these things. Now you can do some things about, you know, how your primer matches to your sequence. And this is more about, like, you know, if we know, for example, we knew where the exon junctions were on that transcript, you could say, hey, make sure that these things are on different exons so they only amplify mRNA and things like that. But there are so many parameters here that you can mess around with, and I don't even want to show you this, but I'm going to down here too. So it's really a matter of, I would say, use the defaults, and then if you have to start tweaking things, you just have to start looking at these and see what gives you the results you want to have. Obviously, in PCR, sometimes you're forced to make primers for sequences that are not optimal. And so in some cases, you just have to pick primers by eye. But this will give you primers that are more likely to work. I hope that answered that.
Great. Another question, and this is a more kind of a basic blast question. They are interested in designing PCR primers, and often a single template gets either selected or an accession gets put in this PCR template box. And the question is, do they need to blast both the sense and antisense or regular sequence and reverse complement, or can they just get away with only using one?
Just one. I mean, I'm not sure quite, I don't quite understand the question. You can only put one strand in here, right? So you put your sequence in here, it designs primers for, it's going to design primers that you would put in this box eventually. There's going to be the forward primer and the reverse primer on your template sequence. So no, you just need to put one strand in here. That seems to be the question.
Yes, that's exactly the question. Okay, cool. One is a little more complicated, but it's a great question. When designing primers for multiple sequences, can you get degenerate primers? A lot of people are interested in finding degenerate primers or using degenerate primers to tease things out. Is that possible currently with the system?
No, it's not possible with Primer Blast. Primer Blast will not work with degenerate primers. I can understand how it's useful. I myself have done that in the past, but there isn't a simple way to do it in Primer Blast. Part of it has to do with the blast search, because blast doesn't work with degenerate codons, or, you know, in places where you use one of those bases like inosine and things like that. So no, it's not possible right now.
Great. Before we go on to more of the direct applications, a person just wrote in recently. So if you have an accession, but you want to just look at a query range, since you're on this page, how would you identify a query range?
Well, so you can't, on this page, you would do that by just putting in the range that you want. Does that make sense? Now this tells you where you want your primers to go on your query. But that's a little bit different than what the person is asking, I think. So you're just going to, you know, in other words, if you don't want to put a human chromosome 1 in here, right? Obviously you're going to have some kind of gene in mind, like you want to get the exon of a particular gene, get that sequence and put it in here. But there isn't a way to indicate the range like you can with BLAST, where the range of the query sequence. You can say where you want your primers to bind, forward and reverse primers, and I do that all the time. So like if you're trying to amplify an exon, you can say, hey, I want my primers to be in this intronic region, you know, outside of that exon. But no, there's no way to specify the range of the template necessarily, except by using these boxes.
Okay, one last question from the same person. Also, if you're wanting to do a sequence search for PCR at the same time, let's say you want to do 2 sequences, can they put in more than one accession and have it run?
Well, can use the option that we talked about today to put more than one sequence in there, but they have to be very closely related to each other. So there's no way to run two independent primer blast searches at the same time. There's no way to do it in batch, except for this feature here where these sequences have to be very similar to each other for this to work. Because it has to find a region. It has to find a region where it can bind a primer where the sequences are 100% identical and match the criteria for designing your primer.
Great, now we have more application questions. One of them is specifically about using primers. What if they happen to have a set of primers and they want to be able to figure out where it would amplify? Is there a way to identify what region would be amplified with a set two primers?
Yes, and in fact, we did that today. We found what region would be amplified from a piece of genomic DNA, but you can just put your primers in these two boxes here, forward and reverse. And then you have to figure out, like, okay, what's my target database, right? Am I amplifying it from a human cDNA library? Well, then this is correct. If I want to be real general about it, I can go here and say something like amplify it from the default nucleotide database, and maybe just say, I mean, probably you have some idea what group of organisms it is. Let's say it's mammals. You can put that in there, and then you can just see what they amplify. Just in exactly the same way with the demo we did today, where we showed that they amplified that region on that primate assembly, the macaque assembly that we did today. So yeah, this is one typical way to use primer blast. What do my primers amplify? The other way, of course, is how can I make primers to amplify this particular sequence that I'm putting in the template box?
Great. I have just two last questions since we're coming up towards the end, and they're both applied questions. And the first question, again, is kind of conceptual. Let's say you have a sequence you've amplified, and you want to identify the organism of that sequence. What should you rely on after you do a blast search? Should it be query coverage, percent identity, e-value? What would you look for to try to identify what that organism is?
Well, yeah, that's a good question, and it doesn't have a single answer, I don't think. It depends a lot on the kind of, I mean, we've done a couple of webinars about this particular topic. And so you kind of want to look, I think probably the simplest answer is you want to look at both coverage and percent identity, those two things together. E-value, I think for identification doesn't really enter into it, right? Because, they're just going to be, they should be just zero. Your e-values are going to be extremely low. So it really depends on, how big your query sequence is versus how big the database sequence is you're matching. So if you're doing like a 16S or something like that, is the database 16S, how big is it? Does it cover the whole thing? Does it cover part of it? Those kinds of considerations. So it requires that you look at both percent identity and the query coverage and how the length of the database sequence relates to the length of your query sequence. So there isn't a single thing, I don't think.
Great. Now, the next question, I think actually could even go beyond BLAST. There's something new. The person is interested in finding orthologs. Are there any new ways or improved ways of finding orthologs?
Well, sure. This is, as you say, this is beyond BLAST, but let's just show them. You saw it in passing when I showed you this. So there's a button right here. So if I have a human gene and I want to know what the orthologs are, I can just press this button right here. And so here are the ones that we've calculated. And you can browse this by group of organisms. Suppose you're interested in fish, killifishes, I don't know. So there's the ortholog in Cyprinodon. Depending upon the gene, you can go beyond this So orthologs, by our definition, are, we know it's the same gene because we know the genomic context, the genes around it, but then if you're going, far away, looking at things that are very distantly related, you can also get this gene similar to TP53, and that's going to take you outside of these the vertebrates here, which is what we've done at this point to the orthologs, so you can see things in other organisms. I'm taking a chance clicking on a link to see if this is going to work. Yeah, so we got all these other kinds of animals in here. So we can see we've got hits to arthropods, flatworms, segmented worms, mollusks, nematodes, in addition to the ones that we saw before where we had chordate sequences, vertebrate sequences anyway. And also we have some jellyfish sequences here, or at least Cnidaria of some kind. Other jellyfish, hydrozoans and anthozoans, corals. So that is the thing that we have now available for getting orthologs or related sequences that's outside of BLAST.
Thank you. We're going to round everything up with a couple of answers to some additional questions that people have had, and this is just general information. Where can we get details of future webinars and register for them? We have an NCBI Insights blog and also several different social media sites, Twitter, Facebook, and LinkedIn as an example. And I've put a link in the chat pod for you so that you can keep an eye and even sign up to let us, to get information on upcoming webinars that we're releasing. Finally, the last two questions that people have asked, quite a number of people have asked about certificates, and the answer is yes. You will get a follow-up email after this webinar, and it will have a certificate that you participated in this webinar. So we thank you very much for your participation and the questions, and we hope you have a very good day.
Thank you, Rana. Thanks, everybody, for coming. Talk to you next time.